Bismillahirrahmanirrahim. Assalamu alaikum. I am Syed Shahabuddin Shah from the Department of Biochemistry, Khaja Azmi University, Islamabad. Today, I am going to talk about the pathophysiological impact of metabolic stresses on human platelets. Metabolic stress or hypermetabolism is a condition which is caused due to the excess or lack of essential metabolic nutrients such as fatty acids or glucose. High fatty acids and high glucose concentration leads to increased ROS production, which ultimately cause metabolic diseases such as obesity and diabetes. High fatty acid concentration is found in obesity, while increased glucose concentration is found in diabetes. Platelets are small, tiny circulatory cells which lack nucleus. Platelets are altered in obesity and diabetes. Platelets perform many different functions. The major function of platelet is the blood hemostasis. When blood vessel is injured, platelet adhere to that injured site and start aggregation and also release cytokines and other molecules. Other than hemostasis, platelet also have role in neuroinflammatory diseases such as Alzheimer's and Parkinson disease and cardiovascular diseases. Platelet also interact with bacteria and leukocytes. When platelet is stimulated with its agonist such as collagen, thrombin, or ADP, the agonist binds to their respective receptors and initiate downstream pathway. ADP, when binds to P2Y1 receptor, it activates phospholipase C, which convert phosphoinositol bisphosphate into phosphoinositol triphosphate and diacylglycerol. The diacylglycerol activates protein kinase C, which increase calcium in the cytosol. On the other hand, phosphoinositol triphosphate binds to its receptor on ER and release calcium from ER into the cytosol. The increased calcium concentration in the cytosol trigger many different signaling pathways and also activate the inside-out signaling and platelet surface integrins. The high calcium concentration in cytosol is buffered by mitochondria, while mitochondrial calcium unipolar complex, which composed of many different subunits. MICU1 and MICU2 are the gatekeepers, and EMRE is the positive regulatory subunit of MCU, while MCUB is the negative dominant subunit of MCU. At low calcium concentration, the MCU channel is closed, while in high calcium concentration, the MCU become open and the calcium goes from the cytosol into the mitochondria. The increased calcium concentration in mitochondria cause increased production of ATP. The aim of our study was to find the pathophysiological effect of metabolic stresses on human platelets with several objectives. Number one, the platelet aggregation in obese individuals and the role of MCU in platelets of obese individuals, and to check the effect of high glucose on human platelets. In this study, we recruited people of young age from age of 20 to 25 years old. To confirm the obese individual, we measure their BMI and waist circumference, and there was significant difference between the BMI and waist circumference of normal and obese individuals. Further, we checked the lipid profile and glucose level in normal and obese individuals. We found that there was significant difference in the triglycerides and VLDL in normal and obese individuals. Then we find the platelet aggregation in normal in obese individuals. Platelet aggregation was measured by time-lapse spectrophotometric assay in which absorbance is measured. The decrease in the absorbance show the platelet aggregation. We found that there was no difference between the platelet aggregation between the normal and obese individuals. Then we checked the acute effect of high glucose on ADP-induced platelet aggregation. For that purpose, we treated the platelets with different concentration of glucose. And our results show that there was no effect of high glucose on ADP-induced platelet aggregation. Next, we check the acute effect of high glucose on platelet count. We treated platelets with high glucose and mannitol, and we found that there was no effect of high glucose or mannitol on the platelet count. As it is known that platelets are hyperactive in obesity, so we hypothesized that MCU might be involved in this process. For that purpose, we measure the expression of MCU and EMRE, the positive regulatory subunit of MCU. Protein were isolated from platelets and Western blot was done to find the expression of MCU, the core subunit, and the EMRE, the positive regulatory subunit of MCU. We found that there was no difference between the MCU and EMRE level between obese and normal individuals. In conclusion, we can say that platelets do not respond in obesity and under acute high glucose treatment. This may be because of the already activated platelets. And MCU and EMRE may not be involved in platelet aggregation or activation in obesity. Thank you.